Today, I want to talk about the role of FADD, the protein normally associated with cell death, and its role in G1 to S cell cycle transition. The cell cycle generally consists of 4 steps. I think you might have all seen this diagram before, so I will not spend too much time on it, but it's important to know that in G1 phase, cell activity is the highest, and it's also where the most cell functions are carried out. It kind of serves as preparation for S phase, which then involves DNA replication. Some cells never go past G1 and become arrested in G1 or enter G0, so they basically never replicate. This is the case normally for highly differentiated cells already. Other cells, of course, go into S phase and then through the whole cell cycle until they end up with 2 identical daughter cells. A key checkpoint here is the restriction point that you can see here in G1 phase. It is regulated by a series of proteins. You might have seen these diagrams before, especially the top one of APCC activity. APCC with CDH1 is a protein complex that together with cyclins and cyclin-dependent kinases controls the progression of the cell cycle. The key point here is that APCC CDH1 depletion is needed to pass this restriction point and enter S phase. As you can see here, this whole depletion of APCC CDH1 is regulated by a series of other proteins. There's kind of a cascade of proteins, which is in turn regulated by oncogenes, of which KRAS is one. You can kind of think of KRAS as a molecular on-off switch for cell proliferation. So KRAS receives signals from the outside of the cell. When it receives the right signals, maybe from other cells, from neighboring cells, or in general from the environment, for example, growth signals, for example, or dividing signals, it turns on this whole cascade of cells until it reaches the depletion of APCC CDH1, which then in turn lets the cell go through this restriction point and transition to S phase and go through the whole cell cycle mitosis and end up with a set of 2 identical daughter cells. This normally works absolutely fine, it is regulated. The problem with cancer though, or with KRAS mutated cancer, is that it is constantly turned on, regardless of if the right signals come from the outside. So this leads to an uncontrolled proliferation of cells, even if the cell is not supposed to do that because it's not ready, because it has damage in the DNA, because it's not big enough, and this leads to problems, obviously. Just to provide a little bit of background for KRAS mutations are involved in many different types of cancer. For example, non-small cell lung cancer, or PDAC, pancreatic ductal adenocarcinoma. PDAC has a 5-year survival rate of about 10%, and this is despite numerous new therapies being developed in the past years. So what we wanted to do in the lab is to investigate how FADD or FADD influences the progression of the cell cycle. For that, we used a genetically modified cell line. In this case, it was H1975. This is a KRAS mutated form of lung cancer. And it was genetically modified, so it appears fluorescent under the microscope, depending on what stage of the cell cycle it is in. And this is due to the concentration of 2 proteins, CD21 and geminin, which are known values of concentration in the cell cycle. So we kind of have a baseline here. So in G1 phase, the cells will appear green because of an accumulation of CD21, and in S phase, they will appear red because of an accumulation of geminin. And in G2M phase, which is the 3rd phase of the cell cycle, it will appear as orange because of a mix of CD21 and geminin. We then use live cell imaging, in which we let the cells under the microscope for 72 hours. It was a controlled environment, so CO2, temperature, everything stayed constant. And in the end, we're left with a reasonably big movie that can then be analyzed using ImageJ, which is basically an imaging software that tracks the fluorescence of each individual cell throughout the 72 hours, gives out data, and then we use MATLAB to basically get the data from ImageJ and create like graphics and model, I guess. So it's kind of a more prefabrication or analysis of the data. The first 3 rows here of the data set represent the control group. So nothing's changed. It's wild type cells. We just let them be, so to say. What is very striking here is that while in the first control set, G1 phase lasts about 5 to 6 hours, which is the normal time frame, I'd say. In the bottom group, where FADD was blocked using siRNA, so FADD was turned off, so to say, G1 phase lasts up to 22 hours, which is a remarkable increase, and it really shows what a crucial role FADD has in the cell cycle. So basically what we've done is we've proposed this new pathway for the depletion of APCC CDH1, in which FADD can also lead to the desired effect and let the cells enter S phase. In terms of next steps, what we want to do now is identify the partners FADD interacts with in the cell cycle, and also investigate the efficiency of different pharmaceuticals that have been developed as CDK4 and 6 inhibitors, the cyclin you can see on the bottom left, such as palbociclib and abemaciclib, alone and together with a block of FADD. So the hope is that together with a turned off FADD, so to say, they have a much better efficiency and lead to a much better outcome and quality of life of patients than right now. To end the presentation, I'd like to sincerely thank the members of my lab that have helped me a lot throughout the past year and from whom I learned a lot. Alamos Rentula, the PI, my mentor, Sergio Lavadia, and Myrna Sito, whom I worked with a lot during the past year. Thank you for your attention.